Good afternoon, everyone. Thank you for tuning in GenScript webinar today. I'm your host, Moon Hua. If this is your first time joining our webinar, here is some background of GenScript. GenScript is the world's leading biotech company providing life science services and products with gene synthesis, peptide, protein, antibody, and preclinical drug development service capabilities, and driven by the corporate missions of making people and nature healthier through biotechnology. GenScript strives to become the most trustworthy biotech company in the world. In today's webinar, we are going to discuss how to speed up and simplify antibody and protein purifications with magnetic separations. So let me introduce our speaker today, Dr. Jane Tan. Dr. Tan graduated with a PhD in biomedical science and worked as a research fellow at the Singapore Institute for Clinical Science. Her research area focused largely on establishing an epigenome for clinical samples and involved collaboration with several government hospitals. This webinar will take around 45 to 50 minutes and it will be a recorded session. If you have any questions during the presentations, you may type your questions in the Q&A box provided, and we will answer them after the presentations. So over to you right now, Dr. Jane.
Okay, thank you so much, Bunhua, for the introduction. So, hi, good afternoon, everyone. Thank you so much for joining me here today. So, my name is Jane, and I'm the field application scientist from GenScript. Okay, so for today's webinar, we will be discussing how magnetic-based separation techniques can actually help you speed up and simplify your antibody and protein purification process. So for any one of you who are interested in purifying either antibodies or proteins, do consider magnetic-based methods for purification because they do have some benefits as compared to the traditional column-based method for purification. Okay, so before we actually talk about magnetic-based technology, first let's look at why are pure proteins required. So pure proteins are actually very useful because they can be used for different applications. For example, you can actually use pure proteins for performing analytical characterization. So say, for instance, if you're interested in determining the amino acid sequence of the protein, then yes, likely you require a very pure protein in order to determine the amino acid sequence. Or if you wish to perform, say, structural studies or functional studies on that protein, then again, yes, you require pure proteins in order for you to really gain insights into its structure and its function. Okay? So not only are pure proteins required in analytical characterization, they can also be used for various applications in various industries. So for example, in the pharmaceutical industry, these pure proteins, for example, antibodies, will be required to be extremely pure when you are going to use these antibodies as therapeutic drugs. Okay, not just that, things like antibodies or enzymes that are used in diagnostic kits will also have to be pretty pure in order for you to get a decent and accurate results from your kits. Even in the food industry, so it doesn't have to only be in the in the pharmaceutical industry, even in food industries where enzymes are actually being used for food processing. So these enzymes do have to be of a certain pure grade as well. And even in industries like in the cosmetics industry, in some cases, you actually have to deliver pure proteins to patients. Okay, so as you can see, right, pure proteins are required in various types of applications. So now that we know why are pure proteins required, let's now see how are pure proteins actually obtained. So if you think about trying to purify a protein from a sample, typically the sample will contain other contaminants. So potential contaminants that may also come with along with your sample, it could be other biological molecules. So for example, you could have lipids or other proteins in your sample. You could have things like debris, salts, reagents. So these are other things in your samples that you do not want. Okay, so you only want your protein of interest. So how do you purify your protein of interest from other contaminants? So you need to identify some difference between your protein of interest compared to the other contaminants which are present. So identify some physical characteristic that is unique of your protein. So you can purify your protein based on that physical characteristic. Okay? And the method that you choose depends on the level of purity that you require. So as you can see here, this is just some commonly used methods for purifying proteins. So you can see you've got centrifugation, which purifies proteins based on density. You've got salting out, which is based on solubility. Electrophoresis, which separates proteins based on size or charge. But generally, I would say these methods do not really result in very high levels of purities because they are not that specific for your protein of interest. And then you've got methods like based on resins. Okay, so these resins could be packed in columns. And depending on the chemistry of these resins, you can separate your proteins based on either its size, its charge, its hydrophobicity, or based on affinity. And of course, you can also use magnetic beads to actually pull out your protein of interest based on affinity. Okay? So these are just some commonly used techniques. But I would say generally that affinity-based methods tend to result in higher purification purities because affinity-based methods tend to be more specific to your protein of interest. Okay, so again, if you require extremely high levels of purities, affinity-based separations is probably the way to go. And again, you can use either resin-based columns or magnetic beads to perform this. Okay, and at GenScript, we actually do have solutions if you are looking at affinity-based purification, whether it's based on resins or based on magnetic beads. So now let's look at the similarities or the differences between resin-based methods for purification versus magnetic beads-based types of affinity purification. So these two fall under the category of affinity-based separation. So the main difference, right, when you are talking about resin-based affinity separation versus magnetic-based is in terms of the type of bead that you will be using for each application. So if you are looking at resin-based type of purification, likely you may be using agarose type of beads for this purification. Whereas if you are using magnetic-based techniques for purification, then the type of bead that is used would be a magnetic bead. Okay, so the beads are very different in both types of purification. So in terms of size, generally agarose beads tend to be a little bit bigger in size, so about 50 to 150 microns in size here. Magnetic beads tend to be a little bit smaller in size, so about 1 to 100 microns, which can be advantageous for magnetic beads because typically the smaller the particle size, then collectively you can have a larger surface area to volume ratio. Okay. And in the case of agarose beads, however, as you can see, they are extremely porous. Okay, so an agarose bead can look something like this. Okay, so you'll see that from the surface of the bead all the way into the interior of the bead, you will see pores. Okay, so agarose beads tend to be highly porous, and because of that, although they can be larger in size, but they do have a high binding capacity because you can see there is a lot of surface area for your target proteins to actually bind to an agarose bead because it's highly porous. Although large, extremely large target proteins may not have access to the inner binding sites, but I would say generally for most proteins, it shouldn't be a problem. Okay. On the other hand, for magnetic beads, you will see that most of the time they are non-porous. Okay, so this is an example of how a magnetic bead may look like. So the blue circle over here is your magnetic bead, so it's non-porous. And on the surface, then you will have your ligands, which are bound onto the surface of the bead, and the ligand will be the ones that will bind onto your target protein of interest. Okay? So because magnetic beads tend to be non-porous, so they may have a slightly lower binding capacity because there are no pores on the beads itself for the target proteins to actually enter and bind into the interior portion of the bead because it's non-porous. Okay, but again, if you think about collectively, because magnetic beads can be smaller in size, so collectively that can still help to compensate for the lower binding capacity of magnetic beads. So I would say generally agarose beads, magnetic beads, they tend to still have pretty decent binding capacities, and it really depends from vendor to vendor as well, how these beads have been uh, bound to the ligands and so on. So I would say generally in terms of binding capacities, it's pretty decent for both cases. But the most important difference between an agarose bead and a magnetic bead is that a magnetic bead has magnetic properties, so it's able to be attracted to a magnet. So if you have a magnet nearby, like this, you will see that the magnetic bead will be attracted and moves towards that magnet. Whereas in the case of an agarose bead, okay, because it is not made of a magnetic material, so even if you have a magnet nearby, it will not move towards the magnet. And this is actually a very important advantage that magnetic beads have over agarose bead. Because they are magnetic, therefore you can easily just use a magnet to perform the purification. Okay, so we will take a look at how this works later on. But first, let's look at how a typical resin-based separation or purification occurs. Okay, so this is the workflow, okay, that you may expect if you are using resin-based columns for purification. So this is, again, not a magnetic-based method for purification. This is based on columns which are packed with resins, okay? So as you can see here, you see this is a column, okay? So this column here, it is packed with resin. So the blue circles that you see here, these are resins which are packed in the column. And then on the surface of the resin, you have your ligand, okay, which will then again bind to your target protein of interest. So this column can be purchased, maybe pre-packed, or you could perform the packing yourself, okay? So this is how a column looks like. And as you can see at the bottom of the column, there is an opening for your samples to flow through. So what you have when you start out your purification process, you have your samples. For example, in this example here, it's a cell lysate sample. So your cells have been producing your proteins of interest, and then you perform a cell lysis to actually release all your proteins. So your target proteins are the ones in pink, okay? So after you have performed your cell lysis, then what is in your sample or what is in your tube here would still be all the cell debris and particulates from performing your cell lysis process. So what you want to do first, before you load it onto the column, is that you want to perform centrifugation and filtration to prevent your column from clogging, because you want to remove any large particulates which may still be present in your sample first before you load your samples onto the column, because these large particulates can go and block this column. And once your column is blocked, then the flow of your samples through the column will be affected. Okay, so once it's affected, then your entire purification process will be affected as well. So you want to make sure that you remove any large particulates first. You have to do this step before you load your samples onto the column. Okay, so once you've loaded your samples onto the column, then your target protein will bind to the ligand, which is on the resin, while the other compounds do not bind to that particular ligand. And then after that, you perform some washes to wash away any non-specifically binding proteins from your resins. And then finally, when you think it's clean enough, then you add an elution solvent that is strong enough to elute your target protein of interest from your resin. Okay, so again, this is what you would probably expect when you are performing a resin-based method of purification. You have to perform this centrifugation and filtration step. So what about if you are performing a magnetic-based method of purification? So again, you start out with your sample, for example, again, a cell lysate sample, okay? But in this case, you do not need to perform that centrifugation and filtration step, okay? So you just have to lyse your cells and then immediately just add your magnetic beads into the sample. Because as you can see in this magnetic-based method, you are not using a column, okay? So you don't have to worry that this tube over here, so you are just using a regular tube like this, okay? You don't have to worry that the tube will be clogged because you are not expecting your samples to flow out of the tube, right? Everything happens within this tube. So you do not have to perform that centrifugation and filtration. You can skip this step immediately add your magnetic beads, is the one in gray over here, to your samples. And then you can see that your protein of interest in this example is the one in green. Okay, so let your beads incubate the sample for a while. So again, remember, magnetic beads are a lot smaller than resins. So what will happen is that these beads will actually be floating in your solution over here, and then it will find and bind to its target protein, so the one in green. So you can see for every bead here, you have a target protein, the one in green, bound onto it. So incubate your beads to your samples for example, maybe 2 hours incubation. And then after you think that that's sufficient, then you expose a very strong magnet nearby. So you can see then your beads, because they are magnetic, then they will be attracted to your magnet, and then along with the target protein. And then what you can do is then remove your supernatant, because this will contain all the other compounds which you are not interested in, and then perform your washes and elution. So finally, when you add your elution buffer, then you will see that it will break the bond between your protein and your bead. Okay, so this is when you can then collect the supernatant here, and this will be your purified protein. Okay, so as you can see, magnetic-based methods can actually help you skip this centrifugation and filtration step, which easily for one sample may take about half an hour to perform. Not just that, but because you do not use any column for magnetic-based separations, so you do not have to perform packing of the column, and you do not need to worry that the column may be clogged up. Okay, so these are some of the benefits that come with magnetic-based separations. So there are some platforms that are currently available to perform either column or resin-based separations versus magnetic-based separations. So if you look at this table here, okay, it's just some of the platforms that are currently popular. So the first three, the ones in orange, okay, the gravity column, the chromatography method, liquid handling systems. So these platforms tend to use resin-based methods for purification. Okay, so these tend to use columns, whereas magnetic methods or magnetic platforms don't use columns. Okay, so the last one over here, which is a system that uses magnetic rods for purification, they do not use columns. So as you can see, right, different platforms have their advantages and disadvantages. So for example, if you look at the gravity column method, okay, so this is a column that you can see here. So the bottom part here, you can see some yellow portion over here. So that's where the resins are being packed. Okay, so with this gravity column, definitely the advantage of this is that it is low cost, because you do not need to use any instrumentation on this method. So everything is just performed manually. But of course, with that, then comes some disadvantages. As long as you are using a column like this, then your samples have to be clarified first. So this means that you need to clean your samples, make sure that they are centrifuged, filtered, to avoid clogging of the column. Okay, so you will see even in chromatographic methods or liquid handling systems where you are using a column, as long as you are using a column, you will see that your samples must be clarified first. Okay, so this again is an additional sample prep step that you need to perform as long as you are using a column. And of course, for gravity columns, not just that, the throughput will be quite limited, because as you can see in this method, what happens is that the user will perform this entire purification step manually and using gravity. So gravity will be used to pull the samples through each column, and that will take a lot of time. Not just that, but because these resins cannot dry out as well, so the user will have to be at the workbench to make sure to monitor the levels of solvents inside each column. Okay, so its throughput is pretty limited. Manual labor is quite tedious as well. Okay, of course, with chromatographic methods, you can try to automate this entire process. Okay, so the pros of using chromatographic methods is that it helps to automate this process. It can also give you options for gradient elution, so this can be quite useful for method development. It can also take in a wide sample volume range as well, okay? And it can be quite flexible, so you can change the system depending on your needs. But of course, again, for these chromatographic methods, you will be using columns on these systems. So again, the same issue will be faced. You need to perform that sample cleanup step before you actually purify your samples. And not just that, these chromatographic systems, they tend to be extremely delicate. So they can be quite sensitive to things like air bubbles or pressure. Okay, so if you over pressurize a column, then the compressed column may have to be repacked, or you may have to change a column. Okay, so it can be quite sensitive when you use these systems. And again, for these systems, typically the throughput is quite limited as well, because you can mainly process only one sample at a time. So if you have more than one sample, or if you have many samples to purify in a day, then these chromatographic methods may be limited. Okay, and the cost of these instruments can be quite high as well. For liquid handling systems, of course, again, one of the advantages is that it helps to automate the process, and the throughput can be quite high. So typically, you can actually purify up to, I believe, 96 samples on this. So you can actually fit 96 well plates over here. And it can be quite flexible as well. Some of the disadvantages is that, again, as long as you're using columns for purification, you need to perform that sample clarification step. And typically, these types of instruments can only process low sample volumes. Okay, so if you have any, larger sample volumes, it may not be able to help you process your sample, and the cost of these instrumentations can be quite high as well. Lastly, this platform, as you can see here, so this is a platform that makes use of a magnetic rod to help you perform purification. So again, this helps you automate this entire process. Throughput is also quite high. You can actually process and put 96 well plates here, okay, for it to process. But again, as you can see in this system, for example, you can only take in small sample volumes, okay? And magnetic beads may seem pricey compared to resins, but later on we will talk about this a little bit more, okay? So as you can see, right, just to summarize the different platforms over here, again, they do have their advantages and disadvantages. I would say the main disadvantages that you see here may be in terms of throughput. So some of them have, again, limited throughput, okay, while others can only process smaller sample volumes, okay? But it is until GenScript, we actually worked with Amgen to come up with a system that is able to improve on the limitations that I just spoke about. Okay, so remember, the limitations that are typically faced by the current platforms could be in terms of limited throughput, and also sample volumes can be quite small for other platforms. But with this system that we actually worked together with Amgen and came up with this system, this is called the MagSA Plus system. So this system can actually help you automate this process using magnetic-based purification methods. And this can actually help you improve on the limitations that we just mentioned. Okay, so with this system, you can, again, like I mentioned, automate the process. But of course, if you wish to do this magnetic purification manually, you can also do so using magnetic beads like this, and also magnetic racks. So you do not have to get an instrument to perform your magnetic-based purification. You can always do this manually as well. Okay, but GenScript, you have the option of either doing it manually or purifying it with an instrument. Okay, and with the MagSA Plus instrument like this, again, because it is a magnetic-based method of purification, you do not need to perform that centrifugation and filtration step. Okay, so this saves you time. No columns are being used on this system, so you do not need to perform column packing and column equilibration. It can also handle up to medium-sized sample volumes. We will talk about this later on. Okay, so it not only processes small sample volumes, but also medium-sized sample volumes, and it can also give you higher throughput compared to things like the gravity column or chromatographic methods. Okay. So before I talk about some of the features of the MagSA Plus system, this is how the system, again, it looks like. So you can see here at the top of the instrument, you can see this is the interface. Okay, so this is where you will program your purification method and tell the instrument what to do. Okay, so this is the interface, and this is touchscreen, so you can actually use your fingers, or you can use this stylus that is provided with the instrument to key in the parameters on this instrument. Then at the bottom of the instrument here, this is where you will put in your samples into the system for it to purify. Okay, so this over here is a door, okay, that you can open this door, and then inside this instrument there are two racks, okay, and you can then put your samples on these racks, and then you close the door, and then you can key in your purification protocol, and then it will start processing your samples for you. Okay, so this is how the instrument actually works. And in terms of the benefits of the instrument itself, again, it is an instrument that can help you automate the purification process if you are looking at purifying either proteins or antibodies, and therefore it helps you reduce your manual labor required. Not just that, because again, it is a magnetic-based method of purification, so it helps you also eliminate the need for centrifugation and filtration, so you save time. Not just that, but as you can see, it also maintains a closed environment to minimize endotoxins contamination. So because your samples will go into this system over here, okay, which is a closed environment, so it does help to minimize any endotoxins contamination in your final purified samples. Okay. Not just that, but if you were to use our MagSA Plus instrument with the magnetic beads that we have, the magnetic beads that we have are highly reusable. Okay, so this means that you can reuse these beads multiple times from purification, one after another, and this can help you lower the overall cost of your purification in the long run. Okay, I will talk more about the beads later on. So in terms of the specifications itself of the system, you can see that again, it can purify up to 12 samples at one go, okay? And each batch takes approximately about 45 minutes to complete, okay? So imagine if within a day, you can probably purify up to 60 samples. And it allows you to use up to 5 buffers for purification. Okay, so remember when you are performing your purification process, you need to add in things like your wash buffers, your elution buffers. So it allows you to add up to 5 different wash and elution buffers. It takes in a sample volume from 1 mil to 50 mils, okay? Later on, we will talk more about this, but even if you have, let's say, for example, 200 mils of sample volume, which you want to purify, you can still use this instrument to purify them. What you have to do first is to reduce your culture volume first before you load it onto the instrument, because the instrument, as you can see, it's only compatible with 50 mil tubes. Okay, so again, if you have any samples which are, for example, 300 mils, what you can do is reduce your culture volume first until your samples can fit a 50 mil tube before you put it onto the MagSA Plus system for processing. And the minimum elution volume of this system is 0.4 mils. It can help you purify up to 80 mix of antibody or protein per sample, and it's also it allows you to save up to 50 protocols. So 50 purification protocols can be saved in the software over here. Okay, so these are just some of the specifications of the instrument. And again, take note, because it is a magnetic-based method of purification, so the instrument does come with strong magnets. So you will see magnets over here. So if you see 3 bars over here, right? So these are the strong magnets which will come with this instrument. Okay. So this is the entire setup of the instrument. Again, this is the instrument itself, and then you've got your on the right-hand side of the instrument, you will see 8 ports like this. Okay, so each port over here will be linked to a buffer bottle. Okay, so the first port all the way to the 5th port, so ports 1 to 5, will be linked to buffer bottles that are used for purification. Okay, so these the first 5 bottles will contain buffers that you will use for purification. So you can either put your wash buffers or your elution buffers in the first 5 bottles. The 6th and the 7th port will be linked to containers that will contain your wash buffers to wash the nozzles. Okay, so on the system itself, there are some nozzles that will be used to pick up your sample or to dispense your wash buffers or your elution buffers. So these nozzles will have to be washed from sample to sample. So you need to also input wash buffers to wash these nozzles. So those wash buffers will go to ports number 6 and number 7. And then port number 8, which is the last port, will be linked to a waste bottle. Okay, so any used wash solutions will then go to the waste bottle over here. So all these things will come with the instrument, so including the tubings, the bottles, and the racks. All these will come with the instrument. In terms of the software as well, it is a very simple software to use. As you can see, when you turn on the instrument, this is what you will see on the interface. Okay, so you can see that there are 5 tabs on the interface here. Okay, so I would say the first 3 tabs are probably the most the tabs that you will use most often. So the very first tab that you will see is called the pre-treatment tab. I won't go into the details of this tab, but basically this tab looks like this. And the main function of a pre-treatment tab is for the user to clean the system. Okay, so you can see there is a fill function here. So you can actually fill the instrument with cleaning buffer to actually clean the system and to minimize endotoxins in the system itself. Okay, so this fill function is to clean the system and to minimize endotoxins contamination. You've also got a UV light over here, which if you were to turn it on, you can actually sterilize the interior of this system. Okay, so this is mainly to clean the system. Then once you think it's clean enough, then you want to perform your actual purification process, then go to the processing tab here. Okay, so this processing tab is where you will key in your purification protocol. Okay, so you can see this is how the tab looks like. And you will see at the top left-hand corner, so this is where you will input the position of your samples that you loaded onto the system, the physical samples. Okay, so you can see here you have, like I mentioned, there are 2 racks in the system. So the first rack is rack A. So rack A has 6 locations where you can place your samples. So A1, A2, A3, A4, A5, A6. Then you have the B rack, so you can again place another 6 samples on the B rack. So in total, you can process 12 samples at one go. And as you can see, then the rest of the interface is where you would key in your purification protocol. Okay, and once you are done keying in the purification protocol, just click on play, and then the instrument will run the purification process for you. And then the very last tab here, okay, is called stripping tab. So what is this stripping tab for? So imagine after you have finished performing your purification, so the instrument has finished purifying your samples. So what happens is that the instrument will alert the user that purification is complete. And then the user will have to go to the instrument and collect the purified sample from the instrument itself. Okay. So once you have collected your purified protein, what is left in the tubes will be your magnetic beads, right? So like I mentioned, magnetic beads that we have at GenScript are highly reusable. So what you can do is to regenerate these beads so that you can store them and use them for multiple times after this purification. So this is what the stripping tab is for. Is for you to clean and regenerate your magnetic beads. Okay, so the stripping protocol, the interface looks very similar. Okay, so you can just program and tell the instrument how you wish to regenerate these magnetic beads. Okay, so again, these are just some of the how the interface actually looks like. And in terms of settings and the system tab, these are mainly used by our engineers. Okay, so you, as the user, you probably will just be looking at the pre-treatment tab, processing tab, and the stripping tab. Okay, so very simple interface to use. And definitely what is important, for anyone who is looking at purification work would be recovery. So if you look at this is actually some data that is obtained by a customer. You can see that these are some antibodies that they tried to purify, and they purified these antibodies on the MagSA system, as well as a typical conventional chromatographic method. You can see that generally, I would say the antibodies are recovered very similarly. Okay, the recoveries are very similar for both. But of course, remember that with the MagSA Plus system, because it's a magnetic-based method for purification, again, you can save time because you don't have to perform the sample cleanup step. And at the same time, you do not need to perform any column packing or column equilibration. You don't have to worry that any column will be clogging up as well. Okay. We also have some data showing you the recovery results of antibodies versus on the MagSA Plus system versus the automated liquid handler. Okay, so again, you can see that I would say recoveries of these antibodies are generally, again, very similar. Okay, not just recoveries are important, definitely your purities are also very important when you perform a purification step. So this is just some gel runs, as you can see here, of purified antibodies, okay, that were purified using the traditional resin-based method versus purifying it on the MagSA device. So you can see that the purities are extremely similar. Okay, so all very pretty good purities of more than 95%. Okay. And these are just some other results that we have in terms of purity. So we did have two different MagSA systems here, okay, versus that of a traditional resin-based method for purification. You can see that generally when it comes to antibody purities, you can see that the purities are extremely high in all three cases, and endotoxins level are all also very similar, okay, less than 0.03 EUs per ml. So apart from that, with the MagSA Plus system, what you will need to run on the system itself is also to use magnetic beads with these the system. Okay, so again, recall that magnetic beads are the ones that actually allow you to use a magnet for purification. So we do have a very wide range of magnetic beads available for both antibody and protein purification at GenScript. So if you look at, if let's say you are looking at performing antibody purification, so these are some of the beads that we actually have. Okay, we've got protein A magnetic beads, we've got protein G magnetic beads, we've got protein AG magnetic beads, we've got protein L magnetic beads, we've got the MAG protein A mag beads. So if you look at all these beads, you can see that we do, for example, if you look at the protein A mag beads, we do have a version with the letters MX at the back. So if you see the word MX at the back, okay, so this means that these beads have a higher binding capacity compared to the non-MAX version. Okay, so same thing for protein G mag beads max. You can see that the binding capacity is actually higher than the non-MAX version. But the main thing that I really want to talk about on this slide is actually the very last item over here. Okay, so this is called the MAG protein A mag beads. Okay, what is so special about these beads is because these beads can be reused for up to 40 times. Okay, so you can clean and regenerate these beads and use them for up to 40 times. Okay, so these beads are alkaline tolerant. So usually what you can use to regenerate your beads will be sodium hydroxide. So you can actually use sodium hydroxide to clean and regenerate your beads and use them for up to 40 times. Not just that, if you take a look at the binding capacity of these beads as well, it's more than 40 mg per mil, which is actually the highest, one of the highest in the markets right now. Okay, so you expect to bind, that these beads can bind to a lot of your antibodies, and at the same time, it is highly reusable. Okay, and so basically, if you were to see the word MAG in front of our magnetic beads, these means that these beads are extremely reusable. Okay, so this is really a very important feature of these beads that we have. And we also have some data on our MAG protein A mag bead. So these are the extremely reusable ones. So if you look at this picture on the top left-hand corner, you can see that we have our MAG protein A mag beads on the right-hand side. And on the left-hand side, we actually have another supplier of beads. And we put them into two different tubes. And then these two tubes of magnetic beads are put on a strong magnet at the bottom here. So at 0 seconds, you can see that all the beads are still in suspension. However, once you leave the beads there for about 6 seconds, you can see that all our beads have been attracted to the magnet, whereas the beads from other suppliers, like supplier B, for example, may be still in suspension, like this. Okay, so this means that our beads actually respond pretty quickly to a magnetic field. And if you were to use other types of beads, you may expect longer purification times because they may respond a little bit more slowly to a magnet. Okay, not just that, we also did some comparison of our binding capacities that we have. So the MAG, again, the MAG protein A MAG beads does have pretty high binding capacities of more than 40 mg per mil. So you can see over here in this data, we have two beads. The one in yellow is the MAG protein A MAG beads. The one in orange is that of a competitor. So as we expose these beads to different concentrations of antibodies, so at 1 mg per mil, 2 mg per mil, 3 mg per mil, you can see that the binding capacity on the MAG protein A MAG beads in yellow, it starts to increase correspondingly. However, the binding capacities on other types of beads, like those in this competitor, for example, actually saturates very, very quickly. Okay. Not just that, we also tested for how the beads react to being washed by an alkali. So you can see here, the MAG protein AMAG beads at 0 cycles, so at a fresh new bead, has binding capacities of close to about 40 mg per mil. And as you use these beads and clean these beads for 40 cycles, you can see that the binding capacity is still somewhat around 35 to 40 mg per mil. Okay, so it's able to maintain that binding capacity up to 40 cycles over here. But what we found also is that beyond 40 cycles of regeneration of these beads, you may experience a drop in the binding capacity. Okay, so therefore, we recommend that you reuse these beads up to 40 times. Not just that, we also tested for protein A leaking from the beads itself. And as you can see, with various uses of the beads, we do see very little leakage of protein A from the beads, which is great. And again, in terms of regeneration of our beads, we also wanted to check and see that to make sure that our beads are clean after regenerating them. So as you can see in this gel run here, so the odd number lanes, so 1, 3, 5, 7, 9, 11. So these are, the samples are actually magnetic beads before regeneration. So after you've purified your samples using these beads, but before cleaning the beads, we actually loaded these beads onto the odd number lanes, and you can see that the beads still do have some proteins which are bound onto the beads, non-specifically. Okay, and then the even number lanes, lanes 2, 4, 6, 8, 10, 12, they contain the magnetic beads after regeneration. So you can see that after you've performed your stripping process of the beads are extremely clean and they do not contain much proteins over here. Okay, so this means that using the default regeneration procedure of the MagSA system, the residual protein on the mag beads can be removed clean, and then you can store it and reuse these beads again. Okay, and in the case if you are looking at performing tagged protein purification, we also do have beads for them. So you can see here, if you are looking at his tag protein purification, we do have nickel charged magnetic beads. If you are looking at GST tagged protein purification, we have glutathione mag beads. If you are looking at biotin tagged compounds, we do have streptavidin mag beads. If you're looking at flag tagged protein, we do have flag mag beads as well, okay, for tagged protein purification. But again, the main beads that I want to highlight on this slide is the MAG nickel magnetic beads. Okay, again, you see the word MAG in front of the magnetic beads. So these means that these beads are highly reusable. So you can see for the MAG nickel magnetic beads, you can reuse them for more than 100 times, and they are also washable with an alkali, NaOH. Okay, and they are also EDTA resistant. So we did some tests to check for how long can we reuse these beads for, And for the MAG nickel magnetic beads, we actually washed them for 200 cycles in this case. And you can see that the binding capacity is still maintained even after 200 cycles. So we recommend that you actually use these beads for more than 100 times. It should be okay. Okay. So again, this is just to highlight, how reusable our beads are. And again, like I mentioned just before I end off, if you happen to have sample volumes of more than 50 mils, because if you remember, the MagSA Plus system can only take in samples in 50 mil tubes. But let's say if you have a sample with volumes that is more than 50 mils, for example, in this case, in this picture here, you can see that this is probably maybe, I don't know, 500 mils of sample here. So what you can do is that you can use these accessories that we have to reduce your culture volume. So for example, this magnetic wand, what it is that each wand comprises of a magnetic rod here and a non-magnetic sleeve outside. Okay, so what happens is you put your magnetic rod in the sleeve. Okay, so the sleeve will be outside the rod, and then you take this wand and you put it into your culture sample. Okay, so for example, this sample maybe it's 500 mils. So what you can do is that because you have a magnetic rod inside, then your beads, which are in the sample, will slowly be attracted to this magnet. Okay, the sleeve outside the magnet. And then, so you just swirl your magnetic wand in the sample to collect all of your beads. And eventually, this is what you will get, right? So then what you do is you take an empty 50 mil tube nearby, and then just put your wand into that empty tube. And then you slowly remove the magnetic rod. And because once you remove the magnetic rod, the beads on the sleeve will slowly just drop off the sleeve and fall into the 50 mil tube. Okay, so that's how you can actually reduce your culture volume, and then after that, still put your samples onto your MagSA Plus system for processing. Same thing for the MAG magnetic block that you see over here. So it looks like this. And so what you do is you can put this block underneath your container. So maybe this container contains about maybe 200 mils of your sample, which has been incubating with your beads. Once your magnet is nearby, then your beads will be attracted to the magnet at the bottom of the container, and then you can just pour out your supernatant and then resuspend your beads in 50 mils or less, and then just put those beads into your MagSA Plus system for processing. Okay, so these are just some of the accessories that we have to help you reduce your culture volume. And so this is actually the complete solution that we have at GenScript if you are looking at performing either manual method of purification or automated methods of magnetic purification. So for manual methods of purification, you can actually consider using racks like these. So these are magnetic racks. So if you have very tiny sample volumes of up to 2 mils, okay, you can actually use these mini magnetic racks. So these racks themselves contain a magnet. And then what you do is your samples will be in these Eppendorf tubes. And then once your beads have been incubated in your samples, then the beads can be put into this, onto the tube can be put onto this rack. And then the rack will then be used to attract the beads, and you can then manually perform that purification. Okay. Or if you have samples that are about 50 mils, then you can actually use a bigger magnetic rack like this. Okay, so this can actually hold bigger sample volumes. We also have a magnetic box like this that can also hold 50 mil tubes. But in the instance, if you do not wish to perform manual purification, if you wish to perform this using an automated instrument, then do consider the MagSA Plus system because again, it can help you increase your throughput, and it also helps to remove your centrifugation and filtration steps. But in the case, if you have larger sample volumes to process, then again, you could easily just use accessories like the magnetic block and the magnetic rod to help to minimize or reduce your culture volume first before you put your samples onto the MagSA Plus system for processing. Okay, so you can actually reduce even up to 1 liter worth of samples and put that into your MagSA Plus instrument. Okay. And we also do have plans to actually come up with another version of this instrument that can actually process even larger volumes of samples at one go. Okay, and it will be probably called the MagMidi system, but this is still under development. Okay. So before I end this presentation, this is just a summary of all the points that we have gone through. So the MagSA Plus system, it is very advantageous because it helps to shorten your purification time and saves you time and labor. It enables you to purify 12 samples at one go in about 45 minutes. Okay. There are no proprietary buffers or consumables that are required to be run on the system. And of course, we also do provide a variety of magnetic beads at GenScript for both antibody and protein purification. So in terms of antibody purification, do remember that we do have the MAG protein A magnetic beads, and these are alkaline tolerant, okay? And therefore, you may wash them with 0.1 molar sodium hydroxide and reuse them up to 40 times, okay? They also have a very high binding capacity of 40 mg per mil. And for tagged protein purification, for his tag protein purification, we do have the MAG nickel magnetic beads that are also alkaline resistant, and they may be washed with one molar of sodium hydroxide solution and can be reused up to 100 times. Okay, they are also resistant to chelators like EDTA and DTT. And again, lastly, if you do have very large sample volumes, for example, 500 mils or 1 liter, you can actually use our accessories like the magnetic wands or magnetic blocks to reduce your sample to 50 mils or less before you load it onto the MagSA Plus system for purification of these reduced volumes of samples. Okay. So yeah, with that, I've come to the end of my presentation. Thank you so much for your time. And yeah, do we have any questions?
Yeah, hi Jen. So thank you so much for your wonderful presentation. So I guess now is the time for us to answer some questions from the floor. So for ladies and gentlemen out there who are listening to these webinars, right? If you have any questions regarding these presentations or regarding our instruments, regarding how to do a better magnetic separations, now is the time for you to type in those questions in the Q&A box, and we will answer them. So let's go to our first questions. Does phospholipids affect the separation efficiency of magnetic purification of proteins?
Does phospholipids affect? No, so basically, for most of our protocol, what you have to do is just lyse the cells, or sometimes you may not even need to lyse the cells, and then immediately to your samples, you can just add the magnetic bead. So we don't really require you to actually remove these phospholipids from the samples first before you can actually purify it on the MagSA Plus system. Yeah, I hope that actually answers the question that you have.
Yeah. So the next questions, which elution buffers are typically used in the MagSA Plus?
So we don't really have any proprietary buffers, but is this for antibody purification?
Let's see. I mean,
So yeah,
it doesn't mentions. Yeah.
okay. So it actually depends on what you are trying to purify. So typically, if you are looking at, say, for example, his-tag protein purification, you will add some imidazole in your elution buffer to try to elute out these his-tag proteins from your nickel beads. But if you are, say, trying to purify antibodies, then you would probably have another elution buffer, for example, a glycine-based elution buffer that you will use. So it really depends on what you are trying to purify. So you can actually use whatever currently used buffers that you are currently using. So you don't have to use anything that is only specific to the instrument. Yeah, but so again, it depends on what proteins or antibodies that you are trying to purify. That would determine the elution buffer that you need to use.
Okay, yes. So another question that is opposite to the previous one. So are there
Mm.
any proprietary reagents that cannot be used with this instrument?
I would say not really, but the only thing that you may want to just take note of is perhaps the salt concentrations that you are using, just to make sure that we do not have any precipitation of your salts. So just make sure that the salt concentrations that you are using in this instrument is not too high. Okay, so anything I would say maybe hundreds of molars worth of salt may be a little bit too high, but I would say generally for most types of buffers, it's actually very compatible with the system. So you don't really have to worry about that. Mainly, yeah, just worry
Right.
about maybe the salt concentration just in case if it's too high. Mm.
Yes. Right. So can normal magnetic beads, not the MAG beads, right, be reused?
So what we actually recommend is that they can be reused, but we would recommend because those magnetic beads they may not be able to be washed with an alkali. Okay, so they may not be alkali resistant. So for those types of beads, because you don't want any sample to sample contamination, so what we recommend is that you use those beads for the same purification of the same protein. So if let's say you have the first batch to be used on purifying this specific protein, then the second round you use it again on the same protein. So this is just to minimize that cross sample contamination. But yes, you can reuse other types of beads, but typically I would say the reusability is up to about 5 times before you may have to use fresh beads again.
All right, thanks. So here is a very long questions for you. So hi, can you clarify the part about the regeneration step, specifically about the multiple bands representing residues? Are these bands specified proteins targeted by the magnetic beads, or are these contaminant?
Oh no, so those are the specific bands that are purified by the magnetic beads. So that's why you see a huge band there because those beads have been used to purify the proteins. So that's why you do see an enrichment of those proteins in the sample. So it's not the stuff that is contaminating the beads. It's actually the protein of interest that you are trying to purify.
Okay. So the next question is which part of this purification protocol is performed manually, since the
Uh,
MagSA is a semi-automated system. Yeah.
Yes. Okay. So the MagSA Plus system is a semi-automated system. So what that means is that not the entire process. Most of it is automated, but there are actually two steps that you will need to perform the process yourself. So the first step is to incubate the beads in your sample. So that has to be done manually. Okay, and then another step that has to still be done manually will be at the end when the MagSA Plus system has purified your samples for you. Then what you need to do is that you go to the system and then collect your purified sample. So that's the two main steps that will have to be done manually. But from the point of when you start your purification, after you have incubated your samples with the beads, put it in the instrument, and then once you start the purification protocol, all the way until purification has ended when you add your elution buffer, then those steps will be automated for you on the instrument.
All right. So the next question is how do we know how much bits to add per sample?
Ah, so we actually do have a magnetic beads calculator. So for every sample that you have, it's a very simple calculator that we have in Excel sheet form. So you basically just key in, what is the concentration of your antibodies, for example, and what beads are you using for purification. So what is the binding capacities of these beads? So just key in some of these parameters into this magnetic beads calculator. And then this magnetic beads calculator will automatically calculate for you how much beads you will require to use to purify a certain sample. So it's a very simple Excel sheet that we have that we will actually provide you if you are looking at using these beads.
All right. So it comes to the end of this webinar. So I guess we will answer the last questions. So is it possible to purify different size proteins in one sample tube at the same time?
Different size proteins. So likely you will as long as so in one purification protocol, you will use the same wash buffers, elution buffers. So as long as you can actually use the same buffers, that those buffers work for the different proteins, it shouldn't be a problem to purify different proteins in one in the same batch, if that's what you're asking. So just make sure that because if you look at the setup of the instrument will be linked to buffer bottles. So those buffer bottles will be used for the entire batch of purification. So as long as the same buffers can be applied to all your proteins and you get good purification recoveries for all the proteins, it shouldn't be a problem.
Okay. So I guess we come to the end of this webinar. So thank you so much, Jane, for your wonderful presentations.
Thank you.
And we hope this webinar is beneficial for your work. I mean, for all the attendees. So thank you so much, and we'll see you again soon. Thank you.
Okay, thank you everyone.